Can you hear me? All right. Thank you again for the support of the ADRC, the REC Scholar. As Kyle told, I am second year for this second cohort, and then I present part of my results that came from my PhD and also my project as a postdoc that is keep work on this, how we can target autophagy to see if we can delay or decrease the age-related cognitive decline. And then we have assumption that one of the hypotheses why this happens is because we have synaptic decline. And let's understand this a little bit. If you think that more synapses, we're going to have a better communication and consequently going to have a better cognition function, for example, memory. If we start losing that, as happened with the aging process, we're going to start increase the risk to have dementia. And the great correlation marker for cognitive decline in Alzheimer's disease is synaptic decline. Then, if we find a way to stop or to delay this age-associated synaptic decline, we might be able to delay the occurrence of dementia and the cognitive associated with that. One of the hypotheses inside of many others that's how this synaptic decline or synaptic integrity disruption happen. It's because as we age, and this is a hallmark of aging and also neurodegenerative disease, we start to accumulate toxic form of protein inside and outside the neuron. And this, in the late road, is going to affect the synapse. And in other words, you're going to start to kill synapses. And as I told you before, if you kill synapses, you're going to have a worse cellular communication and start to have some cognitive decline, some level. Not necessarily have a disease, but we have a synaptic integrity disruption that can lead to some disease, as for example, Alzheimer's disease. And another hypothesis we can associate with this is we do have some organelles, for example, the lysosome, that should be responsible to clean up or to avoid this protein accumulation during the aging process. And this, I'm not talking about 1-2 years, we're talking about decades of events that start this protein accumulation. But as happened to the synaptic integrity, as we age, we have also age-related lysosomal decline in the activity and in the integrity of the membrane of the lysosome. And what we hypothesize is if we can modulate the lysosome, we can avoid protein accumulation or at least delay it and delay the synaptic decline that will end up at the end of this problem. As one analogy, what I can tell is, imagine if we consider the lysosome as a garbage disposal that we have in the sink. What we try to do is to sharpen the blade, not necessarily replace the garbage disposal or put more garbage disposal in the sink. We are just trying to sharpen the blade that we already have over there. Okay, is that feasible to do? As I told you before, one of the hallmarkers of the neurodegenerative disease is the protein accumulation, and then we have, for example, amyloid beta accumulation for Alzheimer's model. When we test a drug that modulates or sharp one of these lysosomal protease called cathepsin B, is responsible to degrade protein inside lysosome in specific pH. When we positive modulate the activity of this lysosomal protease, we clean up accumulation of amyloid beta in mouse model of Alzheimer's disease. In other model of protein accumulation disease, Parkinson's disease, the similar response we have when we positive modulate this with a drug, this activity of this lysosomal protease, we also decrease the levels of accumulation of alpha-synuclein. That is a accumulation that's happened in Parkinson's disease. And also, when we have a model, and we learned that yesterday, that is not a disease or cognitive problem yet, the myocognitive impairment, we have some decline on cognitive function and synaptic function, and also we have some protein accumulation, but again, it's not a pathology yet. We also, when we modulate with the same compound, the cathepsin B activity, We also have some improvement in cognition or delay the decline, and we have some better synaptic integrity. Okay, in other words, yes, it's feasible and it's possible to modulate, to sharpen the blade of lysosomal to avoid protein accumulation and preserve some or rescue some of the cognitive function in models of neurodegenerative disease or just aging. When we submit those samples to some proteomic analysis, as expected, we can see and then we can find we upregulate pathways that is related with lysosome function, protein degradation, and some catabolic process is exactly what we expected for using these compounds that positive modulate the activity of cathepsin B. Other groups has been found also. The cathepsin B could be a mediator of the cognitive health. This specific work, they look for humans and primates, and the level of exercise is positive correlated with the level of the myokine release after physical exercise, that is cathepsin B. In other words, they just found high levels of cathepsin B is positively correlated with the levels of exercise and, of course, positive levels of cognitive function. When they have a mouse model that is knockout for cathepsin B, they can see some benefits from the physical exercise, but they cannot see this at the same level for those who has cathepsin B. Then they indicate cathepsin B as a mediator of this neurogenesis and cognitive function. Okay. In my PhD, I was interested on that modulation, but without drug or without exercise. I was looking if I can find any way that we can modulate this as a diet or as a natural product without use any pharmacs. And then, my PhD, I screened some natural products to see if they have ability to positively modulate lysosomal function and consequently preserve synaptic integrity. And after few years, we found one that is very promising, that is American ginseng. And the idea of this American ginseng is extract from the American ginseng in a specific formulation we was able to access called Cereboost. And this was the one of, among others, that has positive correlation between synaptic markers and positive modulation of the same lysosomal protease that I mentioned before, that's cathepsin B. Then, we use a model of ex vivo cell culture, and then we insult the lysosome, cause lysosomal inhibition, and as expected, you can see in the black bar, we decrease the synaptic integrity by looking for postsynaptic glutamate receptor units. You decrease that when you cause an inhibition in the lysosome. When we pre-treated those ex vivo model with ginseng, we preserve this synaptic decline. In other words, yes, looks like in a model where we have lysosomal dysfunction, ginseng appears, or this extract from ginseng appears to recover, or better, to preserve the lysosomal function and consequently preserve the synaptic integrity. We also did some image analysis. Here's just some examples of like pre and post synaptic markers. And in the middle columns means under lysosomal inhibition, we decrease the density. As more dark, we can see that image. It means we have lower punctate stain for synaptic markers. And when you have co-treatment with ginseng, we preserve that as we saw before. Okay. Now, I want to translate to see if this in vitro model, we can translate this cognitive protection to a preclinical model. Okay, what we did, we treated Fisher female rats with this American ginseng. We mix in their food for 6 weeks, and then we have a model for, not exactly a model, but we have rats from 3 different ages, young, middle age. We assume them as a mild cognitive impairment. I'm going to show why that. And then we did some cognitive assessment to see if we have preservation or not of the cognitive function, and then some proteomic analysis to see what other targets we also might hit with this treatment. Okay, first, to establish the model, we use, as I told, 3 different ages of Fisher female rats, and then we check for some of the cognitive functions. For example, we test for the exploratory ability the rodents has. And as we can see on this tracking, the first day of this maze, they explore almost every place on that maze. And as expected, in the second day, when we put there, they should explore lower because this is part of their nature. If they do not have any motor problem or cognitive problem, is not a direct measure of cognitive function, but give us some indirect response, we can expect a lower activity. And we see that, but we see the lower activity is more evident in the young rodents, less evident in the aged rodents. What is exactly the aging process cause? The second one is a shock-induced memory test, where we put them in a chamber. We measure in the day one how long they take to go to the dark, and just understand the dark is like a safe place to them. We have a gate, we measure the time. We put them in the second day, and then we measure. If they learn, they will go in the second day much faster than the first one. And that is kind of an indirect learning process measurement. We did that, and then we calculate. We can see the day one in the top blue panels. They explore some part of the light side, and then they move, they just go faster in the second day. Who go faster, again, is the youngs, and as the aged, they decrease the time. What that means, they take more time to find the gate than the youngest one. This is a natural process of aging. Next, in the same chamber, after they enter in the day two, we close the gate and we shock them. That is how we say shock-induced memory. And then, what do we expect in the following day? They will avoid the dark. And then in the third day, we put them again. And then if they remember, they will not, or they will avoid as much as they can the dark. We measure that, and then we say this is one indirect measurement of memory. And as expected, the young has the better results than the aged. And then, yes, by just analyze different ages, we can see, even in pre-clinical model, we have age-related cognitive decline. Okay, we submit those samples to proteomic analysis, and we can see there is some downregulation of synaptic integrity and neurotransmitter, some downregulation of the metabolism, and some regulation of the lysosomal membrane as expected. Okay, then we treated those aged rats, with extract for American ginseng for 6 weeks, as I mentioned before. And as expected, you can see the highlighted blue box. We have some improvements in the aged mice treated or supplemented with American ginseng for the exploratory habituation and for the passive avoidance test, that is the shock box. And basically, yes, what we found is we have a delay on the cognitive decline. They are not preserved as the young cognitive function, but we have a delay in cognitive decline when we treated or supplemented them with ginseng for 6 weeks. The proteomic analysis of this gives us some other targets to look for. And the first two columns of this color map say is a young versus aging rats. The second one is MCI, the myocognitive impairment versus aging. And the third one is the aged supplemented with PenQ. And then you can see for protein quality control pathways, for cell growth proliferation, for synaptic integrity, for cellular response to stress, the American ginseng appears to modulate in the opposite direction many of these canonical pathways. What that means to us is, yes, the ginseng or a diet with extract that targets a specific process in the cellular that can avoid protein accumulation and also preserve the synaptic integrity could be a way how we can delay or avoid to have dementia and consequent age-related disease as Alzheimer's disease. My work didn't stop there. I will stop here because of the time, but what I also have, and I hope I will be next year to talk about that, is about how we can use exercise to also modulate similar pathway and also avoid this age-related cognitive decline. This is not a disease model, but we're probably going to move to the next step and try how this supplementation or how the exercise you're going to modulate in models that represent the disease, but at least is a promised result that, yes, we can at least delay this age-related cognitive decline and preserve some of the cognitive function. With that, I would like to say thank you for Kingsley Lab and Dr. Kingsley, where I did my PhD. My PhD was a collaboration between two UNC campus, UNC Pembroke, where I did my work at Bar Lab, and UNC Wilmington, my work at Kingsley Lab, and now I am a postdoc at Dr. Chisler Lab, where I do also some protein quality control analysis in the brain, in the heart. And just so you all know how important slam dunk is, 3 years or 2 years ago, I don't know anymore, I met Dr. Chisler also in the slam dunk for the first time in person, where we talk in the evening, that was last night, in the network section, and then it was the first time I was introduced to him, and there is where I am right now, doing my research, and just to say, please talk with each other, communicate, network, because slam dunk give you a lot of opportunities. With that, the only thing I want you to learn from me, how you say thank you in Portuguese. I'm from Brazil, and then obrigado, and I'm open for any question.
Great talk. So I have I guess a minor question. So I'm curious about the appetite of your mice that are receiving ginseng. So did you look at body weight differences, or did you look at ghrelin to see if maybe they're reduced in weight, or maybe they're a bit hungrier, because that would affect autophagy?
Yeah, thank you. Great question. Yeah, we look for the body weight change, and we do not have any body weights associated with ginseng supplementation. And also we measure or try indirect to measure how much they eat every day during the 6 weeks. And also there's not prevalence to eat more ginseng. In the previous study where we did PADK oral supplementation, Caitlin, it was there in the lab, in Bar Lab, we prepared the PADK with peanut butter to see if they're going to have more tasteful than to eat, but also there's no other body change associated with food except the aging process that you're going to lose some food. But we look for that. Thank you for the question.